has all the information that a cell or an organism will require to function in the long term. That means that this information is really valuable, so you have to keep it as stable as possible. But unfortunately, what happens is that the DNA can be damaged, can be altered physically or chemically by many different agents. During evolution, there has appeared several repair pathways to avoid that. So basically, when the DNA gets broken, there are three possible outcomes. Either you repair faithfully the broken DNA, and that's the ideal situation because you restore the information that you had at the beginning. And the other option is that you are completely failed, or the cell completely failed to repair the DNA. And that's actually not a bad thing because these cells will die. And in a multicellular organism, it doesn't matter if you lose a few of these cells. The really risky part is or the really risky outcome will be if these cells repair this DNA in the wrong way. And what that means is that you will have mutations. You will alter the information on the DNA. And if this happens a lot, you will have what we call genomic instability, and that means that your DNA is not stable, and this is the cause of many rare diseases and a very common disease that is known as cancer. So we will try to understand in the lab what this erroneous repair actually means. And in terms of broken DNA, what we know is that it's basically the selection between different repair pathways. There are two alternative pathways, and the cell have to decide which one to use at a given time, depending on the cellular metabolism. In the long term, what we will really like to know is this balance is important for these diseases I just mentioned, especially cancer, and we can exploit our knowledge to get better treatment to cancer and other diseases.